I'm going to talk about genetic factors. I am the director of the centre, which includes a wide variety of scientific disciplines, right? And clinic, we have obviously the MND clinic at Macquarie, one of the largest clinics in Australia, and we have research from genetics right through to clinical trials. But my trade, if you like, is genetics. So that's what I know best, and that's what I'm going to talk about today. And I've called it Then, Now and the Future, because I'm going to talk about some of the history of the genetic studies informs where we've come from and some of the excitement of the discoveries, particularly the recent discoveries, what we can offer patients now based upon what we know about the genetics. And most of you will associate genetics with the familial form of the disease. And yes, that is true. Most of what we can offer today is around the familial form. But the now and the future, we are really shifting our focus to try and understand genetic factors that contribute or underpin some of the sporadic form of the disease and our hopes for how we can better treat. So I've used this image because this is part of the history, you know, some of the sort of dark times in our understanding of the disease and actually just recognition of awareness of MND. And it was Charcot back in the mid-1800s who first described MND and gave it its name worldwide, the amyotrophic lateral sclerosis. And he made no mention of familial disease at that time. It was a few years later, this Osler described a family that had this condition that was described by Charcot in 1880. It was the Farr family of Vermont in the US. It was quite a large family that had this disease inheriting. And then a few years later, picking up the pace in the awareness and understanding of what this condition was, recognising particularly the variability in the disease. You know, we know there's this remarkable variability. No two patients present the same way. There might be different sites of onset, might be different way that the disease progresses at different ages of onset and different durations of the disease. All of this pointed to a great variability underlying the disease. We fast forward to 1984 when we still hadn't identified any genes underlying this condition, and that's when the Motor Neuron Disease Research Institute of Australia was created. That is the principal funding body in Australia today, funded largely by this association and other state associations. And it sort of flagged some of the increasing awareness of the disease and the growing research that was occurring worldwide. Rebecca mentioned this remarkable what we call the aggregates or inclusions that are within the motor neurons, which are either it's a hallmark pathology. When pathology is done, this is what is looked for, these deposits in your motor neurons. And that was remarkably it was 1988 that was discovered. And that really transformed our understanding of the disease. Okay. And then in 1993, we found the very first gene that caused motor neuron disease. And it's SOD1 or SOD1. And that was in that same family that I mentioned was described in 1880, the Farr family from Vermont. So it took 113 years of research to find that first gene. Okay, 113 years. That's a hell of a long time of gene hunting. And that's when I'd finished my PhD and was launching into my own independent research career, and through various personal reasons decided I'd start on searching for genes in MND. So it took over 100 years to find the first gene, and I bravely, boldly, stupidly, whatever, thought I'd go and find some more. And then in 2004 we did. The Human Genome Project was what transformed genetic studies. And it was soon after we identified the next gene that caused motor neuron disease. So from over 100 odd years, 11 years. Now, there was a critical discovery in 2006 where those clumps of proteins that are accumulating in the motor neuron disease was found to be a particular protein called TDP43. And we found mutations in that gene, the third gene. Okay, can you see where this is going? Look at the years, 100 years, 11 years, 4 years, and bang, the next year we found our next. That's how fast things were starting to move in 2009. And then, as Rebecca mentioned, this remarkable overlap where a small proportion of MND patients also develop or are diagnosed with FTD, and vice versa for their FTD patients. And the discovery of this gene called C9orf72, which is when mutated can cause one or the other or both. And that really transformed what our understanding of the genetics, our capacity to go and now find more genes, and there was this flurry of gene discovery. So this is a similar figure to what Rebecca put up, but what I'm trying to illustrate here is a couple of things. Is you see that from that one gene when I started to all the genes that have been discovered now. Some 25 genes have been implicated. Another thing I wanted to find there is Australian researchers really punch above their weight. In red, you'll see all the studies where have been funded by the MND ROAA, in part funded by this association, other states associations. We've been involved in around about half of the gene discoveries worldwide. I think only the US and the UK can be comparable to our contribution to understanding the genetics of MND. This is a busy slide. A couple of take-home messages I want to show is that in the pie graph you'll see the genes that have been discovered in Australian families. Now, this is a survey of 212 Australian families. Some of these families are very big, include hundreds of individuals. If anybody here has an interest in genealogy, and as you get to a certain age like me, you take an interest in your family tree and you start searching. And then that's what these people have done, and their families are enormous, hundreds of individuals. So we can identify a mutation in around about 60% of those families. In black, around about 40% of families, we do not know what the gene is. And there are some immediate applications, some immediate outcomes for patients. So we can offer symptomatic testing for those who have a family history of the disease. And around about 60-65% of cases, we can offer a diagnosis. We can get an answer. Now, interestingly, around about 10% of sporadic patients will also carry a variant in one of these genes. And this is what we're talking mutations. Mutations is the word in this case. And of course, that early diagnosis, as Rebecca mentioned, is critical to better outcomes. It has transformed genetic counselling. Pre-symptomatic testing can be offered, but of course we need to weigh the benefits and burdens of knowing whether you carry a variant, a mutation that may cause the disease, but for which we know we have little or no effective treatments. But one of the most exciting things for me is our capacity to offer pre-implantation genetic diagnosis for those who may have the disease, they know they have the disease in their family, can choose to undertake IVF and have tested in order to stop the disease transferring to the next generation. And yes, we have babies born within the MND clinic at Macquarie that are what we call MND free. And the way we do that, it can be done blind. So we have a mother or father who might be a member of a family, need not know their genetic status. They can go through IVF and have this testing done blind. So they never know their own status. And we all carry 2 copies of every chromosome. We track the chromosome that carries the mutation, and we have a means there of seeing whether that chromosome has been inherited to the embryo. One on the left there is a fertilised embryo that's been fertilised in an IVF clinic. You can manipulate the embryo and take a very small number of cells and take the DNA out and track the inheritance of that chromosome that has the mutation. And we select the embryo that doesn't, and that's what's transferred to the mother. And that ensures that we have a child born that will not have the disease. It stops the curse, if you like, in that family. But for genetic counselling, it has some real impacts. So obviously, this is a study from a genetic counsellor at Macquarie, Ashley Cook. There are obviously both some barriers and facilitators to those who may want to undertake testing. So some of those that might influence the decision to test might be, it might inform your life choices, how you might want to live your life if you undertake the test. You may want to inform your children whether they should undertake something like the IVF procedure that I just described. Of course, there's a 50% chance of testing negative if you are a member of a family who has that high incidence. Some of those, the barriers to testing, of course, are not wanting it to determine how you live your life. Perhaps wishing to avoid the guilt of passing it on to children. So it's not clear-cut. Genetic counselling is very important in these situations. But what I wanted to illustrate here was it's more than just understanding the familial forms of the disease. There are significant implications for the sporadic forms of the disease. Almost all of the laboratory-based research worldwide is based upon mutations that have been identified in familial cases. We talk about mimicking the disease in the laboratory, making animal models in order to do drug discovery, and all of that is based upon mutations that have come from familial form. And it turns out that the biology of the familial form is the same biology as the sporadic form of the disease. So the same biological mechanisms are underway. We classify biological mechanisms, and we can come up with thousands, including over 10,000 different biological classifications. And it turns out that the MND genes generally just fall into 3. So that tells us, from a laboratory research perspective, they're the mechanisms we should be starting to understand and target if we want to develop therapies. So I'm going to chat now about the search for both genetic and environmental risk factors. I'm moving from mutations, which we know mutation causes the disease or gives you a very strong likelihood of developing the disease, to those genetic factors which we all carry genetic risk factors for all sorts of things. Those who may get to a certain age like me, visit the doctor and he says for a general checkup and he'll say, do you have a history of heart disease in your family? Because we carry variants that predispose us to heart disease, diabetes, etc. The same, we believe, underlie some of the risk for motor neuron disease. And this was a fascinating study that came out just a few years ago and has been extended this year, where one of our collaborators, Professor Amar Al-Chalabi from King's College in London, and his team looked at population registries, primarily from European countries, and looked at the incidence of MND. And they believe it takes it's a multi-step process, and they believe it's 6 steps, 6 hits, if you like, are required throughout your lifetime to ultimately develop MND. This is not a new concept. This is widely accepted in cancer. Cancer is widely accepted to be a multi-hit, multi-step disease. So in those individuals who have a mutation, there we have, I think I've got the right one, yep, mutations. We had SOD1, C9 off, and these are some of the prominent genes. Those mutations can actually account for 2, 3, 4, in fact, with SOD1, it accounts for 5 of those steps. You only need one more step, would be perhaps an exposure to an environmental toxin or some other environmental exposure, or perhaps another genetic risk factor that you have. You only need one more step, and that's why it's so highly penetrant. Without mutation, 6 steps might be individual genetic risk factors coupled with individual environmental risk factors. Okay? So just some concepts around this. A single gene mutation might be enough to simply cause the disease. And in this instance, can you tell me what number you can see? I had a mixture there. 74. Someone else said something else? 71. Okay, so this is obviously there's a test for colour blindness, and there's one, you can have one mutation that's sufficient to cause this. People with colour blindness will see the number 21 normally. There are intermediaries, but so I often put this up, and some people will come later and say, I saw 21, I didn't know. Anyway, so single gene is enough to cause this. But most traits and most diseases require multiple hits, and it might be environmental and genetic. This is something that requires multiple genetic hits. But there are particular genes that make you more susceptible to having this trait. Now, it's not a disease, but your capacity to roll your tongue carries a significant genetic contribution. But if if you can roll your tongue and neither of your parents can't, you don't rush home and say, what happened here? Who got up to something? Because it's neither necessary nor sufficient to actually cause this trait, but is much more likely to generate. So that's where the genetic predisposition comes from. And we see this because there is a group which, remarkably, despite some poor lifestyle choices, seem to reach 100. The Centenarians. They appear to not carry much of the genetic risk that most of us here do. They are able to get away with it because they don't carry all that risk. They don't have those steps, if you like, to developing much of these disorders. Indeed, the Centenarians get away with it, which is it's so true. How often do you see this sort of thing? So it comes back to they have been fortunate enough to either avoid the environmental exposure or not have the genetic risk factors that most of us here do. And that's what we're seeking to identify. We're seeking to identify those genetic risk factors that we all carry, but which ones do we carry that are at risk for motor neuron disease? And we've put together a consortium called the Solstice Sporadic ALS, the Australian Sporadic ALS Consortium across almost all states, where we are recruiting large numbers of MND patients and control individuals, usually family members, and to seek out those genetic risk factors that contribute to the disease. And what we're undertaking in collaboration with, it's an international study, is what is called a human genome project, for MND, and we are sequencing the whole genomes. Many of you may have heard of sequencing the whole genome. There are now these websites where you can go and sequence your genomes, and MND, what's it called? 23andMe is one of them, where you can sequence your DNA to determine, most of it's around genealogy, doing a family tree, but there are sites that will trawl your genetic data to see if you carry risk variants for various diseases. And we've already sequenced the whole genomes of 650 Australian MND patients and contributed that to the international study, which is Project Mine, which ultimately aims to sequence 15,000 sporadic MND patients and tease out those genetic risk variants that I was talking about. And that's come about because the dramatic advances in genetics and the technologies, where the Human Genome Project took about 15 years and cost $3 billion to sequence the whole genome of one individual. We can do the same today for about $1,500. It's in a matter of hours. That's what's transformed genetics and allowed us to actually decide, well, yes, we can take the DNA of sporadic MND patients and see if we can tease out those genetic risk factors that make predispose to the disease. And of course, it's not just MND, but genetic tests can point to the risks of a wide variety of disorders in the population, but we're focusing on, obviously, on MND. But this is the basis of what we call personalised medicine or precision medicine, where you can determine what you are predisposed, what you are likely to develop during a lifetime, and see if we can intervene to prevent you developing these disorders. What is personalised medicine? Well, it's particularly relevant to treatments, and in this case in clinical trials, where, for example, in MND we have, as I said, no two MND patients are the same. The disease will onset at different parts of the body. It will progress at different rates. It will progress in different ways, different ages of onset and different durations of disease. So we're looking at a biological level what seems to be many different diseases that actually ultimately look the same. So the idea of giving one therapy is really nonsense. We need to target the therapies. And in fact, this is one of the reasons why many of the clinical trials have failed, because we have a situation where some patients benefit, some don't, some actually have adverse response. And when you look at the average across a trial, it seems like there's been no benefit. What we need to be able to do is to take that group of patients, take a series of tests, and we want that to be a genetic test, that's what we're looking for, but there are also biochemical tests. Shu will talk about some, Rebecca talked about some earlier, where we can stratify those patients into the groups and then test the drugs on those that are most likely. Now, this is actually a concept that is widely used in cancer. It's particularly particular types of cancer where an individual will be genotyped, is what we call. We type their genes to determine which chemotherapeutic agent they are most likely to respond to. And then also we test as well their capacity to metabolize. So we either do gene tests of enzymes in their liver. So not only determining what drug to give them, but how much. So that's quite widely established in cancer. We want to do the same for MND. It sounds like pie in the sky ideas, but this is actually starting to filter through now. And this is because this was a retrospective study that looked at past clinical trials in MND, trials of lithium. There were 5 studies here. They were all placebo-controlled, double-blind studies, and involved some significant numbers of individuals. Some very large, so in fact, this one was a very large study, very expensive study. All of them, at the end, failed to show any effect. They've said no effect whatsoever. But if we take this approach, was it just no effect because we had all the patients lumped in together? Well, the benefit of hindsight, in the intervening period, one of the genetic risk factors was identified, and it's in a gene called UNC13A. And so they went back and looked retrospectively at those studies. Now, here's the original. If we look, we've got those patients who were given a placebo and those patients who were given lithium. And tracked over 12 months, you can see there's no change in your likelihood for a patient to survive any longer over that period. But once they separated those out into those who carried this particular form of risk gene from those who didn't, clearly there was. So here we have a situation where we did this study to go back to it and say, let's do this test to identify these individuals who actually benefited from the drug. So that's the goal, is to be able to stratify, identify which patients are most likely to respond to a drug. And we're still identifying. That was one of the first. And in the last couple of years, from this very large international study from which we're part of, and we've contributed 650 Australian patients so far, we've managed to pull out 2 more of these risk genes that are contributing to onset of sporadic motor neuron disease that we now hope to be able to stratify patients for clinical trials. So that really sort of brings me to the end of what I wanted to talk about today. The things are looking brighter, but solving this horrible disease requires more than just the genetics, of course. It requires all sorts of interventions. Shu will talk about some, Rebecca talked about some earlier. And we collectively collaborate and integrate all that information into what we call systems biology to try and solve the disease. And that's what we are trying to do at Macquarie. We've created this MND Research Centre. It is Australia's largest centre dedicated to just studying MND. We have over 80 staff and students just working on motor neuron disease. And these are the multiple disciplines. As I said, I've talked to you about the genetics, but we have the protein. We've heard about inflammation today. We have a team working on the inflammation because the neuroinflammation seems to play a critical role in the progression of the disease. Looking at cells, and particularly that link, as I mentioned, between the genetic risk and the environmental exposure. And then research programs to mimic the disease in the laboratory and identify new drugs that we can take back to the clinic. And we've had two clinical trials in the last 2 years at Macquarie as well. So that's all I wanted to say today. So, thank you. I'm happy to take any questions.